Neurotransmission involves the release from a presynaptic neuron of a neurotransmitter, which then can land and bind to a receptor on the postsynaptic cell. The synapse is the space in between. That triggers a series of events. We'll see that leads to changes in ion flow across the membrane. So in contrast to other signaling events we talked about in which the primary signal does not get in this case, actually, the primary signal is the neurotransmitter doesn't get in, but things do get in from the synapse. They can cross, and typically these are ions, and we'll talk about that. There are a variety of protein players in the membrane that are required for the establishment of the concentrations of ions across the membrane and their movement. So this is a close-up view of some of the characters involved. It turns out that resting cells actually have a negative transmembrane electrochemical potential. In other words, it's about minus 60 millivolts across the membrane, with the negative being on the inside of the membrane. So there's more negative on the inside. So how does that arise? Well, it turns out for us, there would be basically two players. There's more than that, but this makes it simpler. That we're going to see there's always a potassium channel open in the membrane, a small amount of these that are non-gated. In other words, that they're always open. But a really big player is actually an enzyme called the sodium potassium ATP Ace. This is an enzyme that takes ATP and drives the thermodynamically unfavorable process. That is, the movement of 3 sodium ions from the inside to the outside, accompanied by the movement of 2 potassium ions to the inside. So that means that we're going to build up potassium on the inside, sodium on the outside, but because of that imbalanced ratio, the inside is going to become more negative because we only move 2 potassiums in for every 3 sodiums we move out. That's going to establish electrochemical gradient across the membrane, in which the membrane is going to have some voltage across it, minus on the inside. Well, this potassium channel also contributes to it, the non-gated one. In other words, it's always open. As we develop a minus potential inside the membrane, think about it now, because we have higher potassium inside, that might want to go outside the membrane because it's higher concentration inside and outside through this non-gated channel. It's always open. But as it does so, it's going to make the inside more and more negative, and that's going to retard the movement of potassium ions across. So these two, eventually an equilibrium will be achieved. These two players are mostly the ones involved, and we're going to end up with a transmembrane resting potential of about minus 60. Again, to higher potassium inside the cell, higher sodium on the outside of the cell. Now what happens? Presynaptic cell is fired. It's going to release a neurotransmitter into the synapse. There it's going to bind a receptor. And in contrast to the other receptors, we're not going to talk about GPCRs right now. This receptor is actually a ligand-gated channel. And what's going to happen in many cases, when the receptor is bound by the neurotransmitter, there's going to be a pore opening here, and it's going to allow for directionality of movement of ions across the membrane. And typically what's going to happen is because sodium is high out here, and because potassium is higher inside, but because it's negative inside, this mostly allows the transfer of positively charged ions. So we're going to get sodium ions coming into the membrane because it's both higher on the outside, sodium ions, and the inside has a negative transmembrane potential, both attracting sodium ions. That's going to lead to this transient small rise and the transmembrane potential is going to become more positive. That accounts for this region, and all of a sudden all hell breaks loose and it goes whoppingly positive. That's because now a new protein channel in the membrane is open that detects not the ligand that was just released in the synapse, but it recognizes the voltage has changed across the membrane. It's the voltage-gated sodium channel. And what happens here is depicted here. It's going to allow the movement of sodium. Now, theoretically, sodium can go either way, and it won't allow potassium movement because it's selected for sodium. Sodium is the smaller ion that could account for it. But because sodium is higher on the outside of the cell, and because the inside is still negative, has a negative transmembrane potential, sodium is going to be driven inside the cell. And in doing so, raises it now to positive values. Well, all of a sudden, if you measure the transmembrane potential, it reaches a point, a top, and then it starts getting more and more negative. At that case, what happens? There's another voltage-gated channel called the potassium channel. Now, we know at this point the inside is positive. in terms of its potential, and there's more potassium on the inside, so lo and behold, potassium is going to leave the cell. That will make it more and more negative on the inside of the cell and eventually get back to the resting potential. But now the distribution of the sodium potassium is not what it used to be, so the sodium potassium ATP Ace will re-equilibrate that and we'll come back to our equilibrium resting potential. So again, this is all gated by a series of channels and of course the prime player, the sodium potassium ATP Ace, which helps establish this imbalance in ions, sodium and potassium across the membrane. There's one other player here that we haven't talked about. This is also a neurotransmitter receptor. It's when a neurotransmitter binds, and this time GABA. It's a neurotransmitter. It opens this as a receptor, but this time transmits negative ions, chloride ions, and we know chloride is also high on the outside compared to the inside. And although it's a little bit negative on the inside, because the chloride is so much higher outside, some of it can come into the cell. And if it do so, it would make the inside of the cell more negative and actually hyperpolarize the cell. It would make it more negative than, let's say, minus 60, and that would have a tendency to make the cell more difficult to fire. That's what we have when we activate neurons. We can inhibit neuron firing by hyperpolarizing the cell, and the main neurotransmitter that does that is GABA. And we can basically, all the mechanisms for firing a cell that typically involve depolarizing the cell. There's many mechanisms that can account for the selectivity of these channels. Here's an example of a sodium channel. If it were open, one could easily see why potassium doesn't get in, because potassium's bigger. You could easily see why chloride doesn't get in, because presumably there'd be side chains here that would repel the chloride and attract the sodium. It's much more difficult to explain how in the potassium channel a smaller sodium doesn't get in. We'll talk about that in a second. Chloride wouldn't have the right electrochemical environment. For chloride to get in and make it a negative anion channel versus the cation channel, that would make sense if the residues attracting the ions here were of the right kind to attract the chloride and repel the sodium and potassium. Again, sometimes we have channels that are just cation channels. They're not selected for sodium or potassium, but just for positive ions. And these are often the kind that we see when we see ligand-gated or neurotransmitter-gated channels. They can allow both the flow of sodium and potassium across the membrane. Many of the gated channels in neurons are very similar in structure. They belong to a common family, the cis loop family, and they often have multimeric structure, often 5 subunits arranged in sort of this pore-like structure. This example in which I have each of these monomeric units consists of a protein which has hydrophobic helices going through the membrane. It's not a serpentine, it's not a GPCR, but it's a little bit like that. And here's an example of a voltage-regulated potassium channel, KV, and this transient receptor potential channel. Likewise, these are very similar. They have these outer loops, and they have an NC terminus. Again, the same side of the membrane, but now they have 6 transmembrane helices going through. Getting into more detail, how can we have a channel that's selective for potassium and not let sodium, which is smaller, through? Well, there are crystal structures. Nobel Prizes have been given to this because they're of importance. Here we have potassium going through its target. This is the potassium channel, and you can see around here there are going to be residues that gate potassium and don't allow sodium through. If we look in closer detail, here's the potassium going through, and in this pore here we have carbonyl groups. These are close enough that they can attract the potassium, but sodium being smaller, they wouldn't form interactions here, and you need some kind of transient interactions to allow the vectorial discharge of potassium across the membrane. If we initially look at it, potassium is hydrated. To get through the membrane, you've got to strip it of waters. Thermodynamically, you have to pay a price for that. However, once you get it inside this pore, no big deal, because now we replace the stripped waters with the basically ion dipole interactions with the carbonyls, and so thermodynamically no big deal for this to move in. Whereas if you had sodium ion here would be highly hydrated, smaller, higher charge density, and you wouldn't make the compensatory interactions with the sodium ion and the carbonyl oxygens. So hence that's the nature of the selectivity filter that allows potassium through but not sodium, which is smaller. The voltage-gated channels are really, really interesting. Let's look at the potassium channel. Here we have, again, we'll have multiple transmembrane helices. And it turns out two of them seem to be voltage sensors. And the S3 and the S4 helix that goes through. And we're going to have, in this particular case, we'll have 6 of those. Each monomer has 6 of those membranes, helices going through. Now it turns out in the resting potential, one of these helices has a bunch of positively charged side chains on it right down here, probably interacting with this negatively charged region inside the cell, because remember, it's more negative on the inside of the cell. So this is the closed channel. Now what happens is, as we start getting more positive on the inside, that's why we're starting to depolarize the cell. Well, one might argue that now there'll be less interactions of this positive region, positive helical region with these side chains, and they would also move conformationally to a different position within the disposition of these subunits. So we get this movement in charge. One could imagine now we have an open channel, especially this makes sense if now we got more positive on the inside. This obviously would make electrostatic sense. And this allows a conformational change which opens up the transmembrane channel. Now remember, we started this whole process out when we had neurotransmitter binary receptor and it suddenly became a gated channel. This is very common in the nervous system. And so how does that happen? Here's an example based from a prokaryote. Cation, it's a cation channel. Remember we talked about that's what initiated the neural signal in the postsynaptic neuron. Here's a crystal structure in the open state. This is looking down on top, and you can see that there's sort of a pore here that constricts as you go into the cell. This is a view of that showing the pore region. Again, this is on the inside of the cell. And it turns out ions can freely get in here, but once they get down here, they have to become naked, and so they have to make interactions here with groups, and the groups down here are typically a glutamate ring. There's glutamates that circle this, allowing easy interactions with the positive ions here. That accounts for the selectivity of these often ligand-gated channels. And then during this process, we get a conformational change on ligand binding, which then allows, again, the vectorial transport of these ions. Here's the selectivity allowing the transport of the ions across the membrane. Now, it can go both ways, but because initially we said the sodium ion is most likely going to be coming in because it's more negative on the inside and sodium is positive on the outside. So we started off talking that the initial event, after we pre-established the resting potential, is the neurotransmitter would bind to its receptor. Conformational change would allow, as we just talked about, that to become a gated channel, typically allowing the flow of sodium in because it's more sodium on the outside and it's negative on the inside, setting up the initial part of this signal. This rise in the membrane potential, then we would follow with the opening of the gated channels that are gated by voltage. Now, these are going to cause an immediate response in the postsynaptic neuron. It will fire, and that signal will propagate down the neuron and eventually stimulate another neuron. But what about if you want a slower response, maybe a longer-term response? Well, that's when our old signaling pathways come in. Some neurotransmitters can bind to a receptor, and that receptor now is no longer a ligand-gated channel that allow, let's say, sodium ions and depolarization of the inside of the cell in the events we just talked about, but rather it's a GPCR. And when it's in there, okay, we get the transmitter bound. It's just like any old ligand bound GPCR. It's going to become effectively a GEF. It's going to allow GTP to bump off GDP, the dissociation of the alpha subunit, and that alpha subunit can do a variety of things. One thing it can clearly do is it can activate adenylate cyclase. In doing so, turn on second messenger production cyclic AMP, which can activate protein kinase A, and that can become an active protein kinase, and then all kinds of things can. Here are two examples. The protein kinase A could phosphorylate a channel and actually cause it to close. And that's what you would want to do potentially to turn down the neural response, which we need to turn down everything. Here's another example. It might actually phosphorylate another receptor and attenuate it, so it can affect not only the phosphorylation by protein kinase A can affect channel openings and closings, but it can also affect other receptors in their ability to actually respond to their ligand. In this particular case, the G-alpha protein might not be able to interact with this because it's phosphorylated. One could imagine that would do all kinds of things to change the response of the cell. Here's another cool example. The G-alpha protein actually can bind. We said that can target proteins like denylate cyclase, phospholipase C. Here it can actually bind to a channel directly and open it or close it or change basically its properties. Here's another interesting example. The cyclic AMP form not only can activate protein kinase A, but there are some channels in which it can actually go and gate the channel. And this is especially true in retinal and olfactory systems in which we have cyclic GMP gating open channels or closing channels. So there's a variety of mechanisms that are used by the cell that we've already studied. Now, all those events would modulate the response of the cell. Let's say you wanted to really modulate it over the long term. Well, of course, what you could start doing is phosphorylating potential transcription factors. Here's an example. Protein kinase A can phosphorylate this CREB, cyclic AMP response element binding protein. And what it can do, phosphorylate it. It can then translocate to the nucleus where it can bind to the cyclic AMP response element and turn on basically gene transcription. The cell becomes something different now. That can really last long term or can have shorter term consequences, but effectively we have a mechanism for memory. So in summary, we establish a transmembrane potential across the neuron using a couple players that allow high sodium on the outside, high potassium on the inside, also high chloride on the outside, and a negative transmembrane potential across the membrane. Initially, events occur when we have a receptor, neurotransmitter binding its receptor. That's a gated channel because of the high sodium on the outside and the minus potential on the inside. Sodium can come in, start the depolarization of the cells. That's basically starts a cascade of events leading to opening of sodium channels, which then have ultimately changed the membrane potential to allow the opening of potassium channels that are voltage gated and lead to eventually the propagation of nerve response. Again, that can even be modulated further by receptor gated inhibitory channels, GABA, which becomes the chloride channel, which makes the cell harder to fire because it brings chloride into the cell, making it more negative. Those responses are instant, but we can likewise have neurotransmitters that have a little bit longer term duration or very, very long term duration by acting through classical GPCRs to produce essentially second messages in the cell, such as cyclic AMP, which can obviously activate kinases, phosphorylating both gated channels. We could phosphorylate other kinds of receptors that are ligand gated in the neuron. Actually, G-alpha protein can actually bind to channels directly and stimulate it, and actually cyclic AMP or GMP can actually bind to channels and gate those as well. Longer term effects are mediated obviously by complex cascades in which kinases are activated, which can lead to activation of transcription factors which go to the nucleus and change gene expression, which can clearly affect intermediate and very long term changes in the cell to promote learning and memory.